Shalom. The BW reporter system enables to study receptor-ligand interactions. It is extremely useful in cases where the ligand of a specific receptor is unknown, or in cases where the experiments with the endogenous ligand are technically difficult.
This method is sensitive and specific to an individual receptor, easy to operate, and reproducible.
Demonstrating the procedure will be Shira Kahlon, a postdoc from my laboratory.
The day before transfection, plate 10 plates with 10 milliliters of 100,000 BW5147 cells and supplemented RPMI. Then place 100 micrograms of the pcDNA3 plasmid in a tube. Add sodium acetate at pH 5.3 to the tube. After this, add 2.5 volumes of 100% ethanol to the plasmid mixture. Then incubate the plasmid mixture overnight at -20 degrees Celsius. 24 hours after plating the BW cells, collect the cells in two 50 milliliter tubes. Then centrifuge the cells at 515 Gs for 5 minutes, and discard the supernatant. Resuspend the pellet in RPMI without additions and repeat the centrifugation. Resuspend the resulting pellet in 1 milliliter of RPMI without additions. Transfer the resuspended pellet to a 0.4 centimeter cuvette on ice. Centrifuge the plasmid that underwent ethanol precipitation. Then wash the plasmid with 1 milliliter of 70% ethanol, and repeat centrifugation for another 20 minutes. Remove all of the ethanol and allow the pellet to dry.